Now, at this time, in the late 50s and early 60s, I thought we could solve the genetic code by isolating many mutations at the same position in a gene and determining what's the parent, what mutations, what codons are related to other codons, and using chemical mutagen and so on. But the code was solved in vitro by Marshall Liebenberg, and our studies really confirmed in vivo what Liebenberg had proven in vitro. And essentially what we did, the wild type at position 211 is amino acid glycine, and we had two mutants, arginine mutant and a glutamic mutant. But we knew that when the arginine mutant was reverted, and you examined the proteins, you either found threonine, isoleucine, or serine. While if you reverted the glutamic acid mutant, you found valine and alanine. Obviously, the codons must be different, and that must explain it. So we did forward and back mutations, and eventually we identified 26 of the codons for 15 amino acids, all introduced at position 211. A lot of work, and it really confirmed what Marshall Liebenberg had deduced in vivo studies. Now, we continued our studies with a protein. I was convinced that we had to complete the sequence, and it took about 7 or 8 years to complete that sequence. A lot of postdocs were involved, and it turned out to be 268 residues, and at the time it was the longest polypeptide to have been sequenced. And in fact, I don't know if it's still present, but in the Guinness Book of Records, this amino acid sequence was listed as being the longest protein to be sequenced. And we also were interested in the relationship between the various genes in the trypophan of E. coli, and one of my graduate students, Ethel Jackson, studied this, and she made the surprising observation. She isolated deletions, and the whole point was to find out which genes produce proteins that interact with given reactions. And she made this unexpected observation that if she isolated deletions which ended beyond the operator, former operator of the operon, and then somewhere later in the operon, that sometimes the expression was even higher than the expression that would be normally obtained, suggesting that there might be some regulatory feature in addition to the promoter operator. At the time, the promoter operator for the trypophan had been characterized by Cohen and Francois Jacob, and Jacques Monod was giving talks all around the world telling that regulation is expensive, and so it's going to be very simple in most organisms. So we believed him and thought that repression would be it. But this suggested that there might be some other regulatory mechanism. In addition, in this time, people were starting to clone plasmid genes and so on, and I collaborated with Herb Boyer and Don Alinsky, who was one of my former postdocs, and we developed plasmid coli-1, which, as you know, it turned out to be a very popular and very common cloning plasmid. So all these were significant contributions, but this was an interesting finding requiring other studies. So these are essentially Ethel Jackson's findings. Ethel produced deletions in the trypophan, and she isolated these two deletions that had one endpoint in the leader region beyond the promoter operator and one in TRPD. And she introduced these mutations into a repressor minus and repressor plus strain, and yes, it's still regulated by the repressor. But the surprising finding was that, in contrast to wild type with no repressor, the elevated expression of the trypophan, these are two enzymes, alpha and beta 2 of the trypophan, these were elevated in both mutants. This suggested that there was something beyond the promoter operator which has regulatory significance. Now, This is what we knew at that trip-opon in time, and it added the regulatory features that we now deduced by examining what the Ethel-Jackson's findings, and also looking at the nucleotide sequence and predicting what the nucleotide sequence might do. And at the very beginning of the operon, there's a promoter operator, and with tryptophan-activated repressor, regulates transcription initiation. But in this regulatory region, there's a coding region we found for small leader peptide, and an untranslated region which can form alternative secondary structures. And we showed subsequently that charged non-tRNA trip was sensed, and there's ribosome-mediated transcription attenuation termination at the site. Now, it took 10 years to figure out all the events in transcription attenuation. We had no idea at the time. It was a new phenomenon. You had no idea how complex and it would evolve. And many graduate students and postdocs had worked on this problem. And of these, Kathy Squires, Bob Landick, and Roberta Coulter, and Bob Fisher are probably best known to all of you. They all made contributions, but it involved many stages and many events and many years. But what was crucial was there was 160 nucleotide leader RNA between the first structure gene and the promoter. And this RNA could form could have 4 RNA segments, here number 1, 2, 3, and 4, which participated in 3 alternative secondary structures. So segment 3 could pair with 4, could pair with segment 4, and that was a transcription terminator. Whenever RNA polymerase synthesized this terminator and it was allowed to pair, transcription would be terminated. But RNA segment 3 could also pair with RNA segment 2 to form an anti-terminator, prevent this from forming, and segment 2 could pair with segment 1 and prevent the anti-terminator form, so this structure is an anti-terminator. Structure 1, 2 also had 2 other roles we discovered. It also served as a crucial transcription core structure, and furthermore, RNA segment 1 coded for a 14 residue peptide with adjacent tryptophan residues. And this is where tryptophan tRNA charging was sensed. So these events were worked out, as shown here. Transcription begins when there's no linear expression. RNA polymerase binds to the promoter. Then the 1-2 structure forms, and RNA polymerase pauses at the end of the leader peptide coding region. While it's paused, ribosome binds at the start codon and begins synthesizing the leader peptide. And then most important is that the moving ribosome releases the paused polymerase. So now both the polymerase and the ribosome are moving on DNA and RNA. When there's adequate levels of charged tRNA trip, synthesis of the leader peptide is completed, and the leader peptide and the ribosome dissociate from the messenger. This leads to the anti-terminator and terminator forming, because that's more stable than the anti-terminator, and transcription is terminated. So that's the event that occurred when an adequate charge tRNA trip. When there's a deficiency of charged tRNA trip, the ribosome stalls at one of the trip codons in the leader peptide coding region. That disrupts the anti-terminator. And the anti-terminator forms, prevents the formation of the terminator, and polymerase resumes transcription. So, what does this accomplish for the bacterium? Well, surprisingly, it allows 600-fold regulation of transcription. Why should there be 600-fold regulation of transcription? It turns out that tryptophan is the most expensive amino acid to synthesize, and so bacteria devise these regulatory mechanisms to only make enough of the enzymes of the pathway when they need the pathway. Repression operates from excess to mild starvation, and attenuation from mild starvation to severe charge tRNA depletion. So this completed our understanding of attenuation in the early 1980s.